Chapter 17, Special Senses. So as we start our special senses lecture, we're going to begin with olfaction. Now olfaction is a fancy word for smell, so we're going to delve into the sense of smell. Now smell is a chemical sense. Now as we work through our special senses, I want you to keep in the back of your mind the who, the what, the where, the when, the why, and the how. These are questions we're going to answer as we get into this lecture. So the human nose contains anywhere from 10 million to 100 million receptors for smell. Again, here's our olfaction. And the olfactory epithelium of the superior part of the nasal cavity. The olfactory epithelium covers the inferior surface of the cribriform plate. We're talking about the ethmoid bone in the skull, and extends along the superior nasal concha. So we can look at a picture and look at the way smell is detected in our body. So we have our superior nasal concha. We have a little patch here, right here, of nasal mucosa. So this mucus patch at the top of our nasal cavity at the cribriform plate, and extending through those olfactory foramina are these olfactory nerves, at least parts of the olfactory nerves. And then there are free nerve endings that are suspended in that mucus layer. Now that mucus layer serves as a medium for chemicals that are dissolved in the air. They're called volatilized. So these volatilized odorants are going to dissolve in that medium, in that mucus, that's going to fire those free nerve endings of those olfactory nerves. Now we can zoom on in. We see the cribriform plate of the ethmoid bone and those olfactory foramina. We see the olfactory nerves moving through, which gives us three different types of cells found in this olfactory epithelium. Number one, olfactory gland. These olfactory glands are making that mucus. The second are going to be olfactory receptor cells. Okay, these olfactory receptor cells are going to be the cells that are detecting those odorants, those chemicals. So this will be number one. Number two, we have supporting cells. Now the job of supporting cells is to do just that. They are columnar epithelium that's going to support these olfactory receptor cells. This third type of cell, number three, are these basal cells. Now basal cells give rise, these are the cells that are mitotically active that replace and give rise to new olfactory receptor cells. And again, we have an accessory structure, this olfactory gland, which is going to produce the mucus for this patch. Above the cribriform plate, we have the olfactory bulb. You'll remember this when we did brain dissection in AP1, where we have our olfactory tracts. So we have all these glomeruli, these little balls of connections from the olfactory nerve to the olfactory tracts. That's going to move to the brain for recognition and identification. So again, there are three types of cells: olfactory receptor cells, supporting cells, and basal cells. So we can look at a histological view at the supporting cells, these columnar epithelium that support those olfactory receptor cell. Here in blue we have an olfactory receptor cell with those olfactory cilia. This is what's detecting those odorants. So in here would be the mucus patch that all those chemicals would dissolve into. So to put it to type, supporting cells are located in the mucous membrane lining the nose. They're used for physical support, nourishment, and insulation. They help electrically insulate the olfactory receptor cells. Basal stem cells undergo mitosis to replace the olfactory receptor cells. And we have our olfactory glands, those Bowman glands that we talked about that are producing mucus that's used to dissolve those odorants, so those odor molecules, so that transduction may occur. Now, as we work through the sense of smell, I want you to recall that we have anywhere from 10 to 100 million different receptors for the various types of odorants. Now, this is unique because our sense of smell is vast. Think of it like a piano. Each key on that piano represents a chemical odorant. Now, when you are smelling pizza, you are detecting hundreds of different chemicals, the chemicals that make up the cheese, the chemicals that make up the bread, the chemicals that make up the sauce, the garlic, the basil. We could go on and on. Now, each of those chemicals represent one key. The reason why you're able to pick out and say, oh, that's basil, or that's tomato, or that's pizza in its entirety is because your brain essentially takes those keys and makes a song. So the ability for our nose to detect these huge variety of odorants, put them together into a concert or song so that your brain can say, ah, this is pizza. So that's pretty unique to our sense of smell. So let's go ahead and follow how it travels. Receptors in the nasal mucosa send impulses along the branches of the olfactory nerve. This is those piano keys. It travels through the cribriform plate, synapses with the olfactory bulb. Those impulses travel along the olfactory tract, and then those beautiful keys are put together to make this melodious song that is interpreted in the primary olfactory area in the cerebral cortex. And this is going to occur in the temporal lobe. Now, our sense of smell is more intricate than we think. Now, while we say the recognition of the stimulant, of the odorant, is detected in the primary olfactory area, which is the temporal region of the cerebral cortex, we do access two other parts of the brain. We access the frontal lobe as well as the hippocampus. Now, why the hippocampus? Why the frontal lobe? The frontal lobe is going to associate that smell. The primary olfactory area detects the stimulant, the odorant, detects the stimuli, and then it goes to the frontal lobe for recognition. Oh, that's pizza, or that is the smell of a rose, or the smell of chocolate chip cookies baking in the oven. The hippocampus is our emotional part of smell. Smell is intimately tied in with emotion. And this is important because when we lose our sense of smell, this is called anosmia. This can happen if, I'll tell you a story, a friend of mine fell off her bike. She hit her head. She went to the hospital because she thought she had a concussion, which she did. And because of the damage and the injury sustained to her brain, she had a temporary loss of anosmia. Temporary loss of smell. She had this anosmia. And as a result, she became depressed. And the reason why she became depressed was because she lost the feeling that she was in touch with reality and everything around her. She could no longer smell the powder in the nurse's gloves when she went home. She actually burned her dinner one night. She couldn't smell the burning food, which, as we know, that burning smell elicits a danger response in our brain. So she felt like she lost touch of reality. So she entered into a state of depression until she regained her sense of smell. So this is important. We intimately tie smells with emotion. So you're sitting, say, in church one day, and the lady next to you has a specific perfume on and remind you of your mother or grandmother, and it can elicit a response. Or we tie in smells with memories, like that smell of those flowers may elicit a response of, oh, that was from my formal in high school, and I had a great time, etc. So we have this unique way that smell is tied into multiple parts of our brain. The next step with regards to smell is to talk about transduction. Now, we introduced this word a few slides ago. Let's define it and really decide and discuss what this means. So olfactory transduction is the binding of an odorant, remember this is a chemical stimuli, an odorant molecule to an olfactory receptor protein, that is that receptor cell. The chemical reaction involving cyclic AMP, CAMP, causes depolarization of that receptor cell. Action potentials travel to the primary olfactory area. Impulses travel to the frontal lobe, orbital frontal area for odor identification. So we can look at a picture to really explain what's happening. So we start with this odorant. The odorant binds to the olfactory receptor protein, which is a G-coupled protein. It undergoes this process with adenylyl cyclase. ATP comes in, converted to CAMP, which opens this cation channel. The channel opens, sodium and calcium move into the cell, depolarizing the receptor potential, causing the action potential to travel to the brain for identification. This is olfactory transduction. The next sense we're going to cover is gustation. So like olfaction, gustation is a fancy word for taste. Now, taste is another chemical sense, so this is the who, but it is much simpler than olfaction. We only have 5 primary tastes that we can decipher: sour, sweet, bitter, salty, and umami, which is this meaty, savory flavors. Now, flavors other than umami are combinations of the other 4 primary tastes. So what does this mean? So we can say that taste is 80% smell. And that's really odd to understand. Let's give it an example. So for sour, lemon versus lime. If you were to plug your nose, essentially stop the sense of smell, all you would be able to taste is sour. Whether you had a lemon in your mouth or a lime in your mouth, all you would be able to taste is sour. That's the sense of taste. But as you're chewing the lemon and chewing the lime, bits of the chemical components travel up into the nasal mucosa, right? And as a result, will fire the olfactory sense and say that this is lemon versus lime. So when we talk about taste, we say that it is not very specific. It is a much simpler type of sense where we rely on our sense of smell to really differentiate sours from different sweets, from bitters and salties. So that's pretty interesting. Now, when we're smelling, we're also tasting. When we're tasting, we're also smelling. So these two senses are intimately tied, one being much more sophisticated than the other, and that being olfaction. Now we could talk about the who's responsible for detecting these stimuli. Now, taste buds are the players that are involved. They contain the receptors for the sensation of taste. Now, with olfaction, we talked about odorants. For taste, we're talking about tastants. Now, approximately 10,000 taste buds are found scattered on the tongue of a young adult, on the soft palate, the pharynx, and even more odd to think, the epiglottis. Now, the function of the taste buds on the epiglottis is a big question mark. We really don't know what they do, what purpose they serve, but they do exist. Taste buds contain 3 kinds of epithelial cells. Well, this sounds familiar, right? So similar cells throughout all our special senses. Number 1, a supporting cell. Number 2, a gustatory or receptor cell. Number 3, a basal cell that is going to give rise to these other receptor cells when they need to be replaced. Let's take a look at a taste bud. So we start with our gustatory receptor cells. This is our first type of cell right here. Right? This is what's leading to these gustatory hairs that are going to be receiving the stimulant, which are the tastants. But the next question is how? Remember, for smell, we needed mucus to dissolve the odorant in order for the receptor cell to detect it. Well, here we need a medium as well. That medium is saliva. So saliva is the medium in which the tastants will dissolve, stimulating those gustatory microvilli, those hairs, which will activate those gustatory receptor cells, sending the action potentials down these first order taste neurons. These little tan guys in between are supporting cells, nourishing, supporting that taste bud. And then way down here, these little triangular shaped cells are our basal cells, giving rise to future gustatory receptor cells. Let's focus in on our taste buds. Taste buds are located in elevations on the tongue called papillae. We have 3 types of papillae that contain taste buds. The first are called valate. So look at that first letter, V. These form a V. In the furthest back portions of your tongue, we have about 12 that contain anywhere from 100 to 300 taste buds. All you have to do is open your mouth, stick your tongue back, look at the back of your tongue. You'll find these enlarged, rounded structures. Those are valate papillae. A lot of people mistake these for glands and think that when you're sick they get enlarged, when in actuality they are taste buds. The next are fungiform. They look like little mushrooms. They are scattered over the tongue with about 5 taste buds each. The third type are called foliate papillae. These are located in the lateral trenches of the tongue, so in the posterior lateral parts of our tongue. Now, most of these degenerate in early childhood. These are really specific for milk, especially when you're breastfeeding as a baby. Now, Now I'm going to digress for a second, talk about babies, and talk about how important their innate sense of smell and taste is, especially as a newborn. Remember, babies can't see very far when they're born, maybe a centimeter or so in front of their face. So how do they know? How do they know where their next meal is going to come from? And when they're first born, the doctor puts that baby on the mother's belly and chest, and what does the baby do? It innately starts suckling and moving its face to the mother's nipple and breasts. And how come? How does it know this? Well, a baby's sense of taste and smell is amazing. A baby can not only smell his or her mother's milk from across a room, 20 feet away. A baby can pick out and smell their mother's milk. Not only that, if you were to do an experiment and give a baby 20 different types of milk from 20 different mothers, that baby can actually taste and decide which milk belongs to his mother. And that's because of these foliate papillae. So that's an amazing feature for children, for babies, to be able to have this sense, this strong sense of taste and smell. And that's because their sense of vision, their other sense, is a little diminished. So as that sense of vision improves, here comes our balance in biology, the sense of taste and that smell will diminish as well. We have another type of papillae. They're called filiform papillae. Now, these guys cover the entire surface of the tongue. So look, here they are. We see these little projections, look like a bristle on a broom, right? And then we get these mushroom-like projections, these fungiform. Now, how come when we talked about taste buds, we said that there's only 3 different types? And this is obviously a fourth type of papillae. Well, here's the difference. This one does not have taste buds. That's why we don't consider this a taste bud, but it is a type of projection, papillae, right? So what are they for if they're not for tasting? They're tactile receptors. That means their job is to increase friction to make it easier for your tongue to move food within the mouth. And where are they even concentrated? Towards the tip of your tongue. If you get something stuck in your teeth, what do you use? You use the tip of your tongue to try to manipulate it out. That friction, that roughness that you feel on your tongue are from these filiform papillae. Go to other animals like cats, tigers, lions, right? Or even the household cat. They have a ton of filiform papillae which give this friction to their tongue. If you ever had your cat lick you and you feel that roughness, almost like sandpaper, that's because of these filiform papillae. So we can put it all together. That V-shape in the back of the tongue, these are our valate papillae, okay? We have our foliate in the lateral trenches of our tongue, our filiforms scattered everywhere, and these little mushroom-shaped guys are fungiform papillae. Notice on the palate, tonsils, and epiglottis, we have more papillae. Now, what nerves are responsible for the sensation of taste? Well, we have 3 cranial nerves that are involved in the sense of taste. They include the facial nerve, which is cranial nerve number 7. It carries taste information from the front or anterior 2/3 of the tongue to the brain. The next is cranial nerve 9. This is our glossopharyngeal nerve. It carries taste information from the posterior 1/3 of the tongue. And then the most important cranial nerve in our body, cranial nerve 10, carries taste information from the taste buds on the epiglottis and in the throat to the brain. We can follow our sense of information from tongue all the way to the medulla, the gustatory nuclei, to the thalamus, to the primary gustatory area of the cerebral cortex. So let's tie it all together, especially with our neuro lecture that we just got finished talking about. We deal with the gustatory pathway from the facial nerve, glossopharyngeal and vagus nerve. Sensation or taste is being detected. It travels up to the medulla oblongata through the gustatory nuclei, where it synapses from first order neuron to second order neuron, moves up to the thalamus, third order neuron to the cerebral cortex. Now that we're done with taste, the next special sense we're going to deal with is sight or vision. So vision uses the visible light spectrum, which is part of the electromagnetic spectrum, with wavelengths from about 400 to 700 nanometers. So again, we're going to do the same target, the who, the what, the where, the when, the why, the how of this system. The The who is going to be who is detecting the stimuli. The what is the stimuli, and luckily we have it right here. It is wavelengths, okay? So the stimuli is going to be visible light. Now, we see the visible light spectrum right here from 400 nanometers is about violet, all the way to 700 nanometers is red. So the length of the wavelengths is going to be important. But then we say that not all yellows are equivalent, not all oranges are the same. So how do we get varying hues of oranges? How do we get varying brightnesses of orange? So we talk about our next slide, which shows a chart, an electromagnetic wave that shows us the number one wavelength, so the length of light, as well as the amplitude. So the amplitude is going to be important because this gives us those darker oranges and then those fluorescent oranges. So to define wavelength, wavelength is defined as the distance between two consecutive peaks of an electromagnetic wave. The longer you get, the more red. The shorter the wavelength is, the more violet. So varying the wavelength gives us all the different colors as depicted by our eye. Before we get into the mechanics of how our eye works, we need to talk about some accessory structures that are here to protect the eye from dust, dirt, water, pathogens, bacteria, etc. We talk about things like the eyelids, eyelashes, eyebrows. The lacrimal apparatus is what's responsible for making and distributing tears over the outer surface of the eye. And then the extrinsic eye muscles, which are responsible for moving the eyeball left, right, up, down, to the sides, etc. Let's start with our palpebral muscles. So these muscles are responsible for controlling eyelid movement, and our extrinsic eye muscles are responsible for moving the eyeball itself in all directions. We have this thin membrane. It's a protective mucous membrane that lines the eyelid and covers the whites of your eye, or the sclera. This is called the conjunctiva. Now, clinically, you might have heard of this. You might have heard of pink eye, right? Or conjunctivitis. So it is an infection of this thin membrane, which causes a buildup of mucus and gunk that kind of, when you're sleeping, closes or sticks closed your eyelids. You have to wake up and kind of free that up to get your eyelids open again. We also have these tarsal plates. These are folds of connective tissue that give form to the eyelids. Now, these tarsal plates are important because they have a row of sebaceous glands. They're called meibomian glands, and these are what is secreting this oil that's going to help keep the eyelids from sticking together. We can look at a picture of the mid-sagittal cut of the eye, and we see a couple things. We see our levator palpebri muscle, so up here, levator palpebri. If you remember muscles from AP1, we talked about these muscles that raise or lift the eyelid. We also have our superior rectus, inferior oblique. The muscles we're not going to be able to see, obviously, because of this cut, is going to be the lateral and medial rectus. Now, we have our eyelashes, again, here to help prevent dust and dirt from getting into the eyeball. We have our eyebrow, which is, again, going to help prevent water, sweat, dust, dirt from dripping down into the eyeball. We have our lower eyelid, our palpebra. Here's our tarsal glands. 1, 2. This is what's going to lubricate the eyelid, again, preventing them from sticking together. We have our cornea, so this tough, fibrous connective tissue. We have another thing that we're going to talk about, the canal schlemm, which is this scleral venous sinus. We'll talk about what that drains in a little bit. Our conjunctiva, 2 bulbar and palpebra conjunctiva. So we see this membrane here, right, as it extends through. The lacrimal apparatus. Its function is to produce tears and then in the same fell sweeping motion drain them as well. So you should be able to trace the pathway of tears from production to drainage. We start with the lacrimal gland, which is in the superior lateral surface of the eye. We have the lacrimal ducts, lacrimal puncta, lacrimal canaliculi, the lacrimal sac, and the nasolacrimal ducts that carry the tears into the nasal cavity. Our next slide will go through a picture so we can trace the flow of tears and look at the actual structures. So let's start with that lacrimal gland. So the lacrimal gland, which is right here, is the structures that are producing tears. So we say, well, what are tears? Well, it's this watery, saline-like solution that is inundated with lysozymes. So again, tears are not only lubricating, wetting the eye, but they also have these lysozymes in there, these enzymes that destroy bacteria or any type of pathogen, dust, dirt that may get in there and could potentially infect the eye. We have these lacrimal ducts. So this is what's going to distribute tears into the eye. Our eyelid is going to sweep those tears over the surface of the eye. So that's why when you're crying or tearing, your eyes blink a lot. From there, we have two holes, lacrimal puncta. These superior and inferior lacrimal puncta. These holes serve as drainage points, so tears drain. Now, from a clinical perspective, if you suffer from dry eye, what an optometrist or an eye doctor can do is they can either temporarily use these cellular plugs that will plug those lacrimal puncta, preventing the drainage of tears. Overall, the effect would be to maintain that moisture in the eye and prevent it from drying out and people suffering from dry eye. Now, they do have permanent plugs as well. They're not cellular based, that can stay in until a doctor removes them. So from those drainage puncta, we lead to the canals. These are lacrimal canaliculi. We have a superior and inferior lacrimal canaliculus. From there, we drain into the lacrimal sac. Okay, now this lacrimal sac will drain into the nose. Now this is why when you're crying a lot or tearing a lot, you get all boogery and your nose starts running, is because those tears drain directly into the nasal cavity. So if you look on the right here, our flow of tears, to reiterate, lacrimal gland secretes tears into the lacrimal extralacrimal ducts, which distribute tears over the surface of the eyeball. Superior and inferior lacrimal canaliculi drains tears into the lacrimal sac, which drains tears into the nasal lacrimal duct, which drains tears into the nasal cavity. Now, we've completed muscles in AP1, so this should be a nice review for all of us. We have 6 extrinsic eye muscles that move the eyes in almost any direction. These muscles include the superior, inferior, lateral, and medial rectus, and then our 2 obliques, or superior and inferior oblique. Now we talk about the actual eyeball itself. It's made up of 2 tunics. Now in Latin, tunics mean coats. There is a fibrous tunic and a vascular tunic. Now the fibrous tunic is going to be the cornea, and the whites of the eye, the sclera. Now just a quick fun tidbit about the cornea. If you've ever poked yourself in your eye, you know how bad it hurts. The cornea has the highest density, the highest concentration of nociceptors for such a small area than any other place in the body. It goes to show you how hard we try to protect this delicate structure. Now for our vascular tunics, this includes the choroid, the ciliary bodies, and the iris, the colored portion of the eye. And we'll get into what each of these structures do serve with regards to the eyeball. So another view of that mid-sagittal cut, so you can identify some of the structures we just talked about. The next slide we're going to look at is a transverse section of an eyeball. Now this is important because we can see some of the more fine detailed structures of the eye. So we're going to start from the superiormost side and we'll talk about the cornea. Now the cornea's function is to act as the eye's outermost lens. Essentially, it acts like a window, and that window is going to control and focus the amount or entry of light into the eye. Now, this is an interesting note about the cornea is that it contributes about 65 to 75% of the eye's total focusing power. So when light strikes the cornea, it bends or refracts, and refraction is going to be important when we talk about focusing that light, focusing that object onto the back of the retina. The next part we're going to talk about is the pupil. The function of the pupil is to allow light to enter the eye so that it can be focused on the retina, and then we would begin the process of sight or vision. Next is the iris. This is the colored portion of the eye, which helps regulate the amount of light entering the eye. Now, just to give you a little example, you've probably seen this. If you shine the flashlight in your eye, you'll see that when there's bright light, the iris constricts, closes the pupil to let in less light, again, ultimately protecting the retina. When there's low light, this iris opens wide open, allowing the pupil to get bigger and let more light in. The next structure that we're going to discuss is the lens. This is essentially a mirror. The lens is again located in the eye, and we're going to talk about cavities and chambers in a little bit. Its job is to change the focal distance of the eye. So it changes shape. The lens physically changes shape. It focuses the light rays that pass through it and focuses it onto the retina, which is eventually going to create a clear image of objects that are positioned at various distances. Now, before we go any further and talk about some more structures, let's talk about some chambers, because we've talked about some structures that are located in the anterior cavity. Now, the anterior cavity, we're going to highlight it here in yellow. So the anterior cavity is made up of the anterior chamber and the posterior chamber. Now, what is in this chamber? Why is it a chamber? Well, it's filled with water, this aqueous humor. Now, remember those canals of Schlemm, those venous sinuses? Well, this is where we're going to drain this aqueous humor. Now, we can talk more clinical connections here, and we'll say, well, what happens if those canal of Schlemms are not draining aqueous humor or enough aqueous humor? What condition does it lead to? And the answer is glaucoma. Now, glaucoma is interesting because you've probably had this test for glaucoma whenever you go to the eye doctor. It's when they do that strong puff of air into your eyeball. What they are measuring is the pressure that's built in that anterior cavity. Now, if the pressure is too high, this can indicate early onset glaucoma or glaucoma. And the problem is that this causes issues on the retina, and it causes issues in the eyeball, can lead to blindness and things like that. Luckily, they do have therapies and surgeries that can hopefully help treat glaucoma. Now, the next structures we're going to talk about in this anterior cavity, which is actually the lens, is the structure that really delineates the anterior cavity from this back portion, this vitreous chamber. Okay, now, the zonular fibers, if you look like they're tied into the lens. Their function is to kind of anchor that lens in place, tie them into the ciliary muscles so that the ciliary muscles can pull and relax on the lens, ultimately changing its shape. The next structure we're going to talk about, since we talked about the ciliary muscles, are the ciliary bodies. Now, on both sides of the eye, we have these two structures that make up the ciliary bodies: the ciliary muscles, again, which pull on the lens, and the ciliary processes. Now, the ciliary bodies, the ciliary processes, are going to make this aqueous humor. Now, that aqueous humor, we make about 3 milliliters a day. Doesn't sound like much, but in the small structure of each eye, 3 milliliters is a pretty significant amount of aqueous humor that is made each day, and therefore recycled through those canal of Schlemms. The next structure we're going to look at is the ora serrata. Now, the function of the ora serrata is to mark the transition from a simple, non-photosensitive area of the retina to that complex, multi-layered photosensitive region. So essentially, this delineates the part of the eye that is picking up those wavelengths and deciphering them to parts of the eye that are not. The next structure, and probably one of the most important structures of the eye, is the retina. Now, the retina is a thin layer of tissue. It covers about 60 to 70% of the back of the eye, and it culminates to the optic nerve. Its main job is going to be to receive light from the lens. It's going to convert that light into neural signals and then ultimately transmit them through the optic nerve to the brain. That's going to be translated into visual recognition. The next structure we look at is the choroid. Now, again, part of our vascular tunics, the choroid is going to provide oxygen and nourishment to the outer layers of the retina. Along with the ciliary body and the iris, the choroid is going to form the uveal tract. The next structure we're going to talk about is the sclera. Now remember, this is one of our fibrous tunics. Now the sclera has this white appearance. It is opaque, it's tough, it is a protective outer layer of the eye. Again, this is the whites of the eye. It is directly continuous with the cornea in front and with the sheath covering the optic nerve behind. Ultimately, the sclera is going to provide protection as well as form of the eye. Now, one of the last structures, well, second to last structures we're going to talk about is the macula lutea. Now, the function of the macula lutea is to be responsible for sharp, detailed central vision. In your lab book, you're going to see that this is called visual acuity. Now, the macula lutea is also called the central fovea or fovea centralis, and we're going to see that this region has the highest concentration of cones in the entire retina. Now, these cones are light-sensitive cells in the retina that are going to give detailed central vision. Again, this is going to be the area of highest visual acuity. Now, the last structure we're going to talk about is going to be this vitreous chamber. Now, the vitreous chamber is going to be filled with this vitreous humor or the vitreous body. Now, it's a gelatinous substance that's going to give the eyeball its shape. Remember, not all eyeballs are created equal. Some are shorter, some are oblong, some are fatter, some are perfectly round. And these differences in eyeballs change the focal point for the fovea centralis, or the central fovea. And this can be problematic. And the reason why this can be problematic is because it can change the way we see objects, which would require accommodations. Those accommodations could be in the form of contact lenses or glasses. So we talked about the iris already. We said that it is the colored portion of the eyeball. And as we can see of the pictures below, it's kind of just elaborating on what we said on our previous slide, that when we have bright light, so again we can highlight bright light. When we have bright light, the pupil constricts, those circular muscles constrict, lessening the size of the opening or the pupil of the eye. This is typically going to be parasympathetic responses. And then with normal light, here we are, we get a nice wide open pupil, allowing normal light to enter the eye and strike the back of the retina. Now, with sympathetic response, fight or flight, we want to let as much light in as possible, allowing for as much visual acuity as we can get. So in dim light, again, this is sympathetic response. The pupil dilates as the radial muscles of the iris contract. It opens everything up, allowing more light. And you can visually see the size of the pupil as we go from 1 to 2 to 3 here. The retina lines the posterior 3/4 of the inner layer of the eyeball. We also find the optic nerve, which is also visible. The optic nerve leaves the eye and causes this weird phenomenon. Where the optic nerve exits, there are no photoreceptors. This spot is called the optic disc, and ultimately it's called the blind spot. And the reason why it's called the blind spot is because of the fact that there are no photoreceptors. So we physically have a blind spot, and you're going to test this in lab, and you can test it at home as well. Now, how come we don't walk around with a hole in our vision? Well, luckily our brain fills in the information for us, so we don't have this weird blank spot as we walk around through our lives. Now, the exact center of the retina is the macula lutea. Within that is the fovea centralis. Again, we talked about this area being our visual acuity area, has the highest visual acuity because we have the highest concentration of cones. This is one of my favorite pictures. This is a view of the retina while using an ophthalmoscope. So I'm going to write this out for you, ophthalmoscope, because this is a device you will use in lab. to examine the blood vessels or the vasculature of the retina. So what can we see here? We can see the optic disc where the optic nerve leaves the eyeball. We can see all the vasculature, all the blood vessels of the retina. And this darkened circle, which you can see with ophthalmoscope, is the macula lutea with the fovea centralis in the center. Again, this is something that we will look at in lab, so be familiar with the device, the name, and what it does. The next step in the process of vision is to examine who receives and interprets the stimuli of light coming in. Well, that is the retina, and it contains our sensors. These sensors are known as photoreceptors. Remember, for taste and smell, we dealt with chemoreceptors. Well, here we have photoreceptors because we are deciphering light. Now, we have two types of photoreceptors, one for dim light and one for color. These are rods and cones. Now, rods are used to see in dim light, and cones are used to produce colored vision. Now, from these sensors, information flows through the outer synaptic layer to the bipolar cells, through the inner synaptic layer to ganglion cells. The axons of these exit as the second cranial nerve, also known as the optic nerve. Now, I'm going to give you a little information about rods and cones that I think is pretty interesting. Now, each eye has about 125 million photoreceptors. Now, we'll talk about the differences between rods and cones, but let's look at their location. First and foremost, rods are usually located around the boundary of the retina, whereas cones are usually located in the center of the retina. Now, look at this little interesting note. Rods make up about 120 million of the 125 million photoreceptors. Cones make up about only 5 million of the photoreceptors. Now, the outer segment is cylindrical of rods, which contains rhodopsin, which is a pigment, and it's made up of vitamin A. The outer segment of cones contains iodopsin, which is another pigment. Now, when we talk about photobleaching, we're going to focus more on rods than cones, and we're going to focus and hone in on rhodopsin. So remember this word as we're going to look at it again in a little bit. Now, remember, rods do not give color vision, and they do not have any differentiation. So here's that word again we looked at in AP1. Now, cones give color vision, and they do have differentiation. They have three types: green, red, and blue. Let's use a picture to look at the microscopic structure of the retina and the direction of light as well as the direction of impulses. So we can start with the neural layer, which is made up of our rods and cones. So we have our cones here and our rods here. This is our photoreceptive layer. Then we get this outer synaptic layer. So those rods and cones, their synaptic terminals are going to touch into these bipolar cells. So that bipolar cell layer is made up of horizontal cells. And you see these cells, their dendrites are going, or their extensions or axons are going horizontally. And then we have these bipolar cells. Remember we said bipolar cells are typically found in the retina, where here they are. They have their axon and dendrites going one way to another. And then we have these amacrine cells, which are major inhibitory synapses on the axon terminals of these bipolar cells. Ultimately, they're going to control and shape the flow of information through the retina. Now, the inner synaptic layer is going to be composed of the axon terminals of this ganglionic cell layer. Now, the ganglionic cell layer, their axons are going to intertwine to make the optic nerve, which will lead into this propagation where the nerve is going to propagate through the retinal blood vessels and all this information supplying nutrients and nourishment to this vascular layer. Now, when we talk about direction of incoming light, light is going to essentially strike here on the retina, and it's going to activate these impulses, right? And it's going to cause a processing of information through these layers until it hits the photoreceptive layer. Once the photoreceptive layer is activated, the actual propagation of action potentials, of electrical impulses, of nerve impulses, is going to move in the opposite direction to the optic nerve, which will move via the optic tracts to the occipital part of our brain, which is our primary visual cortex. Again, another view zooming out at the eyeball, specifically a transverse section of the back portion of the eyeball on the optic disc. So we see the optic disc here. We see all those different layers, and here's that photoreceptive layer, more deep to where the light is going to be striking here at the retina, right? So light is going to come in, it's going to strike, it's going to activate these photoreceptor cells. Action potentials are going to travel this way. Information is going to flow through action potentials down the optic nerve to the brain. Now the next few slides are more review of what we talked about in our previous slides, so just putting it to type. The eye is going to be divided into an anterior chamber and a posterior chamber by the iris, the colored portion of the eyeball. That's what's separating those chambers. The anterior chamber is delineated as being between the iris and the cornea and is filled with an aqueous humor, this clear watery liquid that's made by those ciliary processes and ciliary bodies. The posterior chamber lies behind the iris and in front of the lens and is also filled with aqueous humor. Behind the posterior cavity, we have the vitreous chamber or vitreous body, which is filled with this gelatinous substance that gives the eyeball its shape. Now, light passes through the cornea, passes through the anterior chamber, the pupil, the posterior chamber, the lens, the vitreous humor, and is ultimately projected onto the retina. Now think about that. We just got finished saying that there's all this water inside the anterior and posterior chamber. Then you have the actual cornea, that tough fibrous connective tissue protective layer. Then you have the lens. Then you have that gelatinous structure before you get to the retina. So this leads to an interesting dilemma. We have something called refraction. So when we'll draw, say, water here, let's draw an eyeball. It's not a great eyeball, but we'll draw an eyeball here, right? So when you look at an object in the water, we'll draw, say, a fish. Again, my drawings are not that great, but we have a fish. When you're looking at that fish, it appears to be right here, when in actuality, the fish is here. So what does that mean? What's happening? Light refracts in water. Okay, so when you're looking at something in the water and you're looking straight down, you have to deal with refraction. So light bends. That's essentially what we're saying. So if you want to catch that fish, you're going to cast your line not here, but way in front, because you're accounting for that refraction. And this is a trick that the properties of physics play with our eyeball, and our eye is no different. We're going through a whole bunch of mediums: water, gelatin, lens, connective tissue. So our eyeball, our retina, has to account, and the lens is going to help account by focusing the object and accounting for all that refraction to get the object directly onto the central fovea. So again, we talked about the vitreous chamber in the anterior cavity. So again, just a review, zooming on into the anterior cavity. But what I do like is we can see the canal or Schlemm pretty good here, where the flow of aqueous humor through the ciliary bodies into that anterior and posterior chamber, filling it with water and then draining. Remember, making about roughly 3 mils of aqueous humor a day. Table 17.1 is a great table because it goes through all the different structures of the eyeball by highlighting them for you and giving their function. For example, the fibrous tunic. It's made up of, 1, the cornea, 2, the sclera. What does the cornea do? It emits and refracts or bends light. The sclera provides shape and protects the inner parts, right? So we go to the vascular tunic with the iris, the ciliary bodies, the choroid, the retina, its job the lens, anterior cavity, and posterior cavity. So it gives you a nice coloration to help explain what everything is. So again, just a repeat, puts everything that we talked about in the past few slides into a nice little neat package. Here's that theory behind the refraction of light. So this is what we have to deal with in our eyeball. This is what I drew on the other page. So light refracts or bends when it passes through a transparent substance with one density into a second transparent substance with a different density, like our lens, our aqueous humor, and our vitreous body. This bending occurs at the junction of the two samples, or the two substances. So you see here's light coming in, and then once it hits the water, it bends to a different direction. Which brings us to another issue. We're moving objects, well, light, right? The rays of light through a lens, essentially a mirror, and through water. Now, what does this mean? When you look in a mirror, everything is backwards, right? And if you look through a microscope, everything is upside down and backwards. Now, our eyeball is no difference. Images are focused on the retina, are inverted, and right to left reversed because of this refraction. Now, luckily, our brain corrects the image, so when I look at you, you're not upside down and backwards, but you're standing correct and left to right correct. Our brain does this for us and does it immediately, which is amazing if you think about it. The lens must accommodate to properly focus the object. Remember that image is going to be projected onto the fovea centralis. This is the site of highest visual acuity. Let's look at an example. So what happens when you look at a distant object versus a close object? How does your eye know? Look out your window. Look at that tree. Based on its refraction, it's going to be the same size to your eyeball as the pen sitting right in front of your face. So how does your brain decipher that that tree is not the same size as the pen or smaller and is actually a huge tree versus that pen is a size in front of you, a normal size of a pen and not a huge tree? Well, this goes through experience, but it deals with something called accommodation. Our eye has the ability to accommodate, and you could do this. You can measure your visual accommodation. Take your pen, put your arm fully out in front of you, and slowly bring that pen towards your face. Now, a couple things are going on, right? Number one, the size of the pen appears to be changing, but it's really not. We know the pen's staying the same size. Number two, these zonular fibers are getting more loose. As those zonular fibers loosen, the shape of the lens changes, which is accommodating for the object moving closer, keeping it focused onto the fovea centralis. So again, this is called near point accommodation. Now we look at a distant object, we see its focal point, it is being focused onto the lens. The lens is now focusing it onto the fovea centralis. Now, think of it like this. If you ever played with a magnifying lens as a kid and you had the light come through, the sunlight come through the magnifying glass, and then it projects a pinpoint that you would put on a piece of paper to, burn that piece of paper. That's essentially what your lens is doing. It's taking that light and making a pinpoint spot, a focused pinpoint spot on the back of the retina. Now we're able to account for distant objects that are distantly far away or ultimately closer by changing the tautness on these zonular fibers right here. Now the next question is, what happens if your eyeball is misshapen or you lose the ability to change the shape of the lens? Now a normal eye is called the emmetropic eye, and it will refract light correctly and focus a clear image onto the back of the retina. So images coming in, it focuses directly onto the back of the retina. This is a normal eye, and we call this 20/20 vision when you use a visual acuity chart or Snellen's eye chart, and you will use this in lab. Now, some of you may be nearsightedness. We call this myopic eye or myopia. Now, what happens is your eyeball is actually misshapen. It's longer than it should be. And as a result, the images converges in front of the retina. Now, these people who suffer from nearsightedness can see objects pretty closely, can see close objects, but have a problem seeing further objects that are further away. So how do you fix this? Well, glasses or contact lenses. And the lens has to be a concave lens, which is used to correct the vision. Let's look at our next slide. And we can see what happens. Look at this eyeball. It's much more longer than a typical eyeball. So this dashed line represents a normal eyeball, right? And we see the focal point way in front of the retina in this longer eye. So what do? We need to put a concave lens, which will change the focal point, putting this focal distance right back onto the back of the retina. Now, opposite to nearsightedness gives us farsightedness or hyperoptic eye or hyperopia, also known as hypermetropia. The eyeball is shorter than it should be, which means that the image converges behind the retina. Now, people who suffer from farsightedness can see distant objects clearly but have difficulty with close or nearby objects. Now, a convex lens is used to correct this abnormality. So this side shows us the farsightedness shape of a hyperoptic eye. So uncorrected, look at this really short eyeball. This is where a normal eye would be. The object is focused behind the retina. So let's use a convex lens. It refocuses the object, changes the focal point, puts the object right back on the retina where it belongs to be. The last condition we're going to discuss is an astigmatism. It's a condition where either the cornea and or the lens has an irregular curvature. Astigmatisms cause blurred or distorted vision. And this happens because light rays are prevented from meeting at a common focal point. Now, clinically, it is treatable by medical professionals. It does require medical diagnosis, and we see that it's pretty common. More than 3 million U.S. cases per year for an astigmatism.